Okay, so we're back on track. Is everybody there? I want to see your faces now again. Okay, so I want to see your faces because I want to ask you who works or who has worked with zoogeochemistry. Just raise your hands up, please. Oh, there's only 2 of us. Okay. Okay. Well, that was because some problems. Well, okay. So let me see. I'll wait till the speakers have joined it. Okay. So who's gonna speak today? We have Elizabeth Leroux from Denmark. Hello, Elisa. We have Judith Sitters from Belgium and the Netherlands. We have Amanda Subalowski from Florida in the US. Jonas, and you're gonna help me with your surname, Jonas Schoelink. Is that correct, Jonas?
Well, just say Jonas, that's okay. No, the surname is Schoelink.
Schoelink, okay. And then me from my lounge in the Netherlands. Right, so first, before we go into a quick quiz that we have prepared for you, Lisa wants to announce that we have beers and nibbles later on. Yes, Lisa?
Yeah, so I thought it would be easiest to just put it on my background so you can have all the information and I don't have to worry. So it's at 5:15 program time this afternoon. And we know we're not going to finish today. So it would be really cool if we could just have a very informal get together, bring a beer, and hopefully make the beer like animal themed, just for fun, and bring an animal themed background. And we're just going to chat zoogeochemistry and also, look at what kind of interest we have in forming a zoogeochemistry community, but more about that in a bit.
Okay, so make sure you got your beers and your background ready, or your wine or your mojito, whatever. Now we have prepared a quick quiz to evaluate your previous knowledge on zoogeochemistry. We're not gonna tell anybody, okay? So it's just to get you familiarized with it. I just stick at the link to the chat, so you can copy it from there or click it. Click it probably is gonna be slower, or copy it and paste it on your browser, and you will be directed to a questionnaire. It's just 2 minutes. We have 2 minutes for that. So in 2 minutes we reconvene, and I'm gonna try to deal with some technical problems, and in 2 minutes I'll tell you to stop. Okay? So be quick. Right. Is that enough? Yes? Yeah. Everybody finished? Yeah. Okay. Then let's go to. We're gonna go first to see the presentation about savannas from Lisa and Judith. So I'm just gonna share the video with you, and then it's gonna be a 15-minute video. I hope you like it. They have prepared a really cool presentation. So let's see. Let's go into the savannas.
Maybe it would be good for the speed of the video to switch off our cameras again.
Yes, of course.
In the vast savanna, one grass fights for survival. Bridled by low soil nutrient conditions and plant-like immobility, she toils endlessly, investing every ounce of energy and reaching with every root fiber towards the distant and gradually depleting soil nutrients of her immediate surroundings. Day by day, season by season, she waits for her one true impala to boldly go where some impala have gone before, but not many, and bring her the nutrients she so desperately needs. But the savannah is a harsh and dangerous place, with menacing corners, where vegetation is dense and visibility is low, and the sense of dread is tangible. And alas, chivalry is not a trait of the fittest. And so the impala, paralyzed with fear, go poop somewhere else. So she waits in vain, dreading the coming growing season as she watches the slow and tragic dwindling of her nutrient supply. But hope was not abandoned, for this was a reserve with mega herbivores, large, fearless beasts that forage in the face of danger, beasts of carefree defecation. Unperturbed by the possibility of peril, predators be damned. This was defecation without borders. And so, with her nutrient stores replenished through a perfectly placed poo pile, our plant protagonist found herself perfectly poised to prosper.
Right. So hopefully that little teaser made you curious enough to sit through the rest of Judith and my presentation. My name is Elizabeth LaRue, and Judith and I want to just walk you through some of the work that we've been doing over the last couple of years. And we've created this mind map to give you an impression of how our work and each story fits together and how one question led to the next. And our intention is to present a very broad overview of our work, so we'll need to skip a lot of details, but we'll be happy to discuss these during the beer and nibbles session. Broadly, we are interested in how consumers influence nutrient landscapes and ecological stoichiometry, but specifically also how species differ in these functions and what species diversity means for nutrient landscapes and stoichiometry. So we focus here on three mechanisms through which species may differ in how they influence nutrient recycling, through differences in vulnerability to predation, differences in the ratios in which they require nutrients themselves, and differences in the ratios in which they consume nutrients. And each of these mechanisms can be linked to animal body size, but also at times to digestive physiology and to feeding types. So through the first mechanism, species differ in their relative vulnerability to predation. Now, predation risk should vary spatially because some predators use structure in the landscape, such as vegetation density, to plan their ambush. And the success of ambush predators is determined by how close they are able to get to their prey without being noticed. So predation risk should therefore be linked to visibility. And visibility varies spatially. And so it creates this landscape of fear to which we expect some species should be responding to more strongly than others. And generally speaking, small species tend to be more vulnerable to predation, and so we expect smaller species to use the landscape according to this variability and risk, whereas larger species should be using the landscape more evenly. And the outcome of this study was that we indeed saw that the smaller species were congregating in these less vegetation dense, presumably safer areas with high visibility, but they were depositing huge amounts of fecal nutrients into these areas. But the larger animals, they used the landscape of fear a lot more evenly. And by doing that, they moved nutrients into dense, risky areas of the savanna. So effectively, the larger animals created this countercurrent in nutrient movement that went against this fear-driven gradient of nutrient accumulation. It also showed that the fecal nutrients that were deposited in the open areas were predominantly from smaller species, whereas the fecal input to the more dense areas were predominantly from larger animals, as we showed in the trailer. So it meant that the differences in the response to predation pressure to some extent determined the identity of the defecator. So the question then becomes, if you have similar amounts of fecal material that's accumulating in these different areas, does it matter who is actually doing the defecating? And the reason why we might suspect that it would matter who's responsible for the defecation is because species are likely to differ in their stoichiometric demand for nutrients, so the ratios in which they require these nutrients. And they would therefore also differentially recycle them. Again, this is also expected to differ along a body size axis. Nitrogen is a key component in protein, so we might expect that the demand for nitrogen should increase with body size in the same way as metabolic rate increases, and this is with an exponent of 0.75. So per unit body mass, smaller animals will require more nitrogen than larger animals. And phosphorus is a key component of bone, so we might expect that the demand for phosphorus should increase with body size in the same way as skeleton size increases with body size. And this is with an exponent of 1.1. So if this holds true, then we would expect that species nitrogen to phosphorus stoichiometric requirement would decrease as body size increase. So with these expected changes in nutrient requirement, what remains in the dung would then be expected to change like this, with fecal nitrogen to phosphorus ratios increasing with body size. And we expect this stoichiometric curve because both fecal nitrogen and fecal phosphorus is expected to decrease with body size, but the decrease in fecal nitrogen should slow down at larger body sizes, whereas the decrease in fecal phosphorus should speed up. And this is because of these exponents of below 1 for nitrogen requirement and above 1 for phosphorus requirement. And we found some very nice support for these expectations in a South African savanna, where the fecal nutrients that were measured in the dung of different sized species followed these hypothesized patterns. So fecal phosphorus declined strongly with body size. Fecal nitrogen declined, but the relationship was less certain. But because of the strong fecal phosphorus change, fecal nitrogen to phosphorus ratios increased strongly with body size.
However, different patterns with body size were found in 2 other African savannas, suggesting that more factors are driving fecal nutrient stoichiometry. For example, Stadner and Elser predicted a quadratic relationship between fecal phosphorus and body size when including smaller animals, and this is because smaller animals have higher growth rates and thus would have a higher metabolic phosphorus requirement. And this would explain the quadratic fecal phosphorus pattern in Tanzania and why the same pattern was not seen in the South African data that only included much larger species. Also, differences in diet stoichiometry are likely to play an important role. And these differences in diet stoichiometry can be linked to the herbivore's feeding strategy. In other words, is the herbivore a browser or a grazer? In African savanna, browsers consume tree leaves that have higher nitrogen concentrations than grass consumed by grazers, and we therefore expect their dung nitrogen to phosphorus ratio to also be higher. And we found support for these expectations in the Kenyan and Tanzanian savanna, where nitrogen to phosphorus ratios from tree leaves were higher than from grass leaves. As a result, browser dung also had higher nitrogen to phosphorus ratios than grazer dung. And this too may explain some of the differences we see in the data from the South and East African savannas. As the South African species set contained hardly any browsers, whereas the East African species sets were more mixed. So, with the expected variation across body size, feeding type, and diet, Lisa and I realized that we could need a data set that contained more species to yield a better understanding of the drivers of fecal stoichiometry. And therefore, we decided to reach out to other researchers working on fecal nutrients of African herbivores to assemble a comprehensive database of fecal nutrients. And so far, we have collected data of 44 herbivore species from 10 different countries. And our first result from this larger data set shows that fecal nitrogen to phosphorus ratios follow our predicted pattern if we take the phosphorus requirements of both small and large herbivores into account. So smaller herbivores have higher metabolic phosphorus requirements, while larger herbivores have higher bone phosphorus requirements, both resulting in higher fecal nitrogen to phosphorus ratios. We also see that the fecal nitrogen to phosphorus ratio decreases with fecal delta 13C. And fecal delta 13C reflects the relative proportions of C3 trees to C4 grass consumed. So this means that the higher the delta 13C value, so in this case the less negative the value, the more a herbivore is grazing. And as we expected, the fecal nitrogen to phosphorus ratio decreases with more C4 grass in the herbivore's diet due to a decrease in forage nitrogen. While the next step in our story focuses on the impact of all this variation in fecal stoichiometry on plants. Plant productivity in savannas is commonly limited by nitrogen and phosphorus, and variation in fecal stoichiometry can change nutrient availability to plants. And as plant species have different nitrogen to phosphorus requirements, variation in fecal stoichiometry might change competitive interactions between plant species. So in an African savanna, we expect that grazers such as zebra, eating grass and producing dung with a low nitrogen to phosphorus ratio will promote the growth of tree seedlings. And this is because many trees in savanna are able to fix nitrogen from the atmosphere. And under low nitrogen to phosphorus supply, they are not constrained by phosphorus availability. Browsers such as giraffe, producing dung with a high nitrogen to phosphorus ratio will promote the growth of grasses, which have an advantage under more phosphorus-limited conditions. And this hypothesized concept thus implies that browser dung will promote grass growth, while grazer dung will promote tree growth. A greenhouse experiment where we grew plants in pots as monocultures or mixtures with dung from either zebra or giraffe confirmed our predictions. The competitive outcome between tree seedlings and grasses was driven by variation in fecal nitrogen to phosphorus ratio. Under zebra dung with a low nitrogen to phosphorus ratio, tree seedlings performed better than grasses, as their competitive strength in this graph is above 1, because they were able to fix nitrogen from the atmosphere. Under giraffe dung with a high nitrogen to phosphorus ratio, grasses suppressed nitrogen fixation and tree seedling growth, thereby winning competition from the tree seedlings. Of course, these predictions now need to be tested under field conditions. So to conclude, a diverse species set in African savannas contributes towards both homogenizing and diversifying the nutrient landscape. Species that vary in vulnerability were able to distribute nutrients more widely, but variation in body size and feeding type ensured that nutrients were distributed in different ratios. In this way, herbivores that occur in diverse communities shape the nutrient landscape that is available to plants, and thereby they contribute towards maintaining diversity within plant communities, and potentially may even help maintain the tree-grass balance which is so characteristic of African savannas. Thank you very much for listening, and we are ready to answer any questions that you might have.
Very, very nice presentation. Huh? Thank you very much, Lisa, and thank you very much, Judith. Fantastic. But we're going to have to wait for the questions. The audience is going to have to refrain and wait a little bit because we're tight on time. So we're going to leave the questions for the end. Okay, I'm going to play the video from Amanda, who's gonna talk about how wildebeest affects the Serengeti, the Mara River, and for how long. Just watch the video. I'll share it. Let me see. There we go. Your fantastic video, Amanda, online. Okay, let me see. Just once again. It's not allowing the resolution. Let me see if I can. Technical problems here. Right. So what I'll do is I'll share Jonah's video about hypos first, and then I'll try to solve the issues with Amanda's video, okay? So Jonah is gonna talk also about how the Mara River is affected by a different vertebrate, a different mammal. Let's find out which.
Masai Mara National Reserve, southwest Kenya. Shaped by volcanic activity millions of years ago, it's home to many magnificent animal species. All these wonders of nature wouldn't be here if it weren't for the lifeline of the Masai Mara, the river Mara. It springs high up in the Mau escarpment and mouths 400 kilometers later in Lake Victoria, the largest tropical lake in the world. Unfortunately, increased land use and agricultural activity are polluting the Mara. Due to irrigation and rainfall, pollutants from fertilizers find their way into the river. We call this agricultural runoff. Downstream, the water of Lake Victoria changes into a green thick soup dominated by blue-green algae. High concentrations of these algae are toxic to both man and animals. Normally, unicellular organisms called diatoms are dominant in lakes as well as in our oceans. Diatoms are a key source of food and energy for other organisms, and they turn sunlight into chemical energy through photosynthesis. In this way, diatoms produce a quarter of Earth's oxygen globally. Human activity and the resulting eutrophication are putting a strain on diatom growth, and thus on the entire ecosystem. To research this, a team of the University of Antwerp went to Kenya. Their colleagues of Yale and the Carey Institute of Ecosystem Studies already set up camp deep in the savannah. Learning from the local people, the scientists have since long built up a thorough knowledge of this ecosystem. In their research, they will be focusing on a particular chemical element, silica. Ever cut yourself on a grass sheet? Big chance silica was involved. The surface of leaves can sometimes feel like sandpaper. This is caused by phytoliths, structures in grass that contain silica, making them less attractive to grazers. Silica is important because on the one hand, the supply of natural silica to rivers and lakes balances unfavorable human nitrogen and phosphorus inputs caused by agricultural runoff. On the other hand, the supply of fresh natural silica is vital for the growth of diatoms. A unique feature of diatom anatomy is that they are surrounded by a shield made of silica. When diatoms die, these shields sink to the bottom of the lake. A lot of silica is thereby lost. The formation of new diatoms thus depends on how much fresh silica is supplied to the lake from land through rivers. But how does it find its way into the water? The team will collect soil and water samples along the riverbank, as well as grass and soil samples higher up the riverbank. Analyzing these samples will help in understanding what processes are going on. To collect the samples, the team gets the help of local park rangers. The Mara River has one of the largest remaining hippo populations in the world. About 4,000 hippos are resident. They prefer to lumber around their favorite spot and are keen to protect it. To collect the samples, the scientists often must find a path through the soggy mud. Carcasses and skulls of wildebeest that didn't survive the Great Migration a few months ago are serving as stepping stones. Once back in base camp, samples are processed the first time in an improvised field laboratory. Water samples are filtered and soil samples are packed vacuum. This should keep all samples fresh for transportation to Belgium. Here, specialized labs will analyze the nature and concentration of the silica in all the samples. The final results exceed scientific expectations. The team discovers that hippos are almost predominantly responsible for strong increases of silica transported along the Mara River. At night, the hippos come out of the water to graze in the savanna plains, often even a few miles away from the river. Giants like hippos don't mind the sandpaper-like structures in plant and grass. Each hippo consumes up to 25 kilograms of fresh grass each night. Once digested, the hippos will already be back in the water, hanging around their favorite spot. In order to mark their territory, hippos will twist their tails while literally spreading their dung around. The phytoliths in the grass they ate are not digested, but once in contact with water, they easily dissolve. Their feeding pattern and specific behavior thus have a large impact on the functioning of this aquatic ecosystem. In a strange way, their seemingly dirty behavior has a big impact on the supply of fresh silica to the river, balancing out human input of nitrogen and phosphorus and supplying fresh silica to Lake Victoria, enabling the formation of precious new diatoms. Conserving these wonderful animals may very well be of crucial importance for keeping healthy the aquatic ecosystems and all the people that live from it.
Okay, so we're back from another fantastic story from Jonas involving the transport of silica from the grasses outside the riverbanks to the river and its impact on diatom production. So very clear the transboundary effects between different types of ecosystems. I would like to ask just quickly, Amanda, if we have technical problems with Amanda's video. So I don't know, she offered to give the presentation live, but I don't know if you are able to do it now or you.
But we can also, we can give her permission to share her screen if she can then show the video from, if you have it on hand, Amanda.
Yeah, I do. Yeah, that'd be great.
Okay, because there's also, so if you share your screen, so you should have permission, and then if you click on share screen, then there should be a under the advanced tab, you can upload the video, so you don't have to stream from your computer, basically. I mean, so it's not just a sharing screen. I guess you did that.
I'm sorry, could you say that again?
So if you click on share screen, and then you go to advanced settings, and then there should be a file that you can upload, then it will be faster. So also we're told. Yeah.
To like actually share the recording.
Exactly.
I don't see where it's letting me upload it.
Okay, maybe then in the interest of time, we just share the screen like regular.
Okay, well, thank you. I will I will do my best to kind of give this off the cuff here. But first of all, thank you all for organizing this session. This has been really, really cool to see this work kind of put together. And we originally organized this so that my presentation would go before Jonas's to kind of introduce the system a bit. And this is based on work that was done in the Mara River, the same river where Jonas just talked about work on silica cycling. But I'm going to be talking about this other phenomenon that happens in the Mara River, which is the annual migration of the Serengeti wildebeest herd. And there are about 1.3 million wildebeest remain in the largest remaining overland migration in the world. And the wildebeest come up from the southern Serengeti plains during the dry season in search of kind of greener pastures and forage. And while they're there, they cross the river multiple times as they move between their dry season feeding grounds. And these are these really kind of exciting, visual events. They're highly photographed. And occasionally there's the kind of individual mortality due to, you can see these lurking Nile crocodiles kind of hanging out in the bottom. And this is an important part, of course, of the life history of the wildebeest and the zebra, but it also plays an important role for the Mara River, because when animals may either defecate or urinate while crossing the river or get killed and die in the river, that results in a resource subsidy from the terrestrial system into the aquatic system. So many ecosystems are influenced by resource subsidies, which are resources that originate in a donor ecosystem and then are moved into a recipient ecosystem and alter the dynamics of a consumer in that recipient system. And aquatic systems in particular can be heavily influenced by resource subsidies because they kind of tend to fall low on the landscape and they have a concave shape. And so things just congregate in aquatic bodies. But animals can also move resources around the landscape, and animal vectored resource subsidies are particularly interesting, I think, for several reasons. One, they're often high in limited nutrients. So animal resource subsidies are usually carcasses or excretion or egestion, which we've all just kind of learned about, how rich those can be in nitrogen and phosphorus. Animals move, so they move resources at faster rates along gradients or even against natural gradients. So think of like fish swimming upstream or animals moving things upslope, whereas otherwise all of those resource subsidies are usually kind of washing downslope. And animals tend to aggregate in time and space, so that can result in hot spots or hot moments of biogeochemical cycling. But there's a lot of context dependency when it comes to the ecosystem effects of animal resource subsidies, because it combines the influence of characteristics of the animal vector, like body size, and in the donor system, so what they're grazing on, and things like resource stoichiometry. And then those inputs are filtered through the lens of recipient system characteristics. So how big is the system that they're moving those nutrients into? Is it already limited in any kind of nutrients? And what is the stoichiometry of that resource availability? So altogether, these things kind of interact to ultimately determine ecosystem effects. So we've been studying this in the Mara River, which runs from the mountains in Kenya through the Masai Mara National Reserve and the Serengeti National Park and into Lake Victoria. It's the only permanent source of water in this kind of huge conserved ecosystem. So it's a really important resource, both for wildlife and for about 1 million people that rely on it for surface water. And because there are so many large animals still maintained in this part of the world, and in particular in these protected areas, it's a really interesting place to study the influence of large wildlife on resource movement on the landscape. So as I mentioned, the wildebeest move from the Serengeti up into the Mara, and they spend a portion of the year up here. And they cross the river multiple times during this period. And often, as you saw, the crossings are more or less successful. Maybe a few individuals die due to crocodile mortality, but sometimes they cross the river at a really bad location, either a steep exit, really rocky where the rocks get slippery, or the river is particularly high. And this can result in mass drownings of hundreds or thousands of wildebeest in a single crossing. And these are actually fairly common events. So with anecdotal observations from guides and lodges and rangers, and from our own surveys going on about 10 years now, we've documented the occurrence of mass drownings in 18 out of 20 years, with an average of over 6,000 carcasses entering the river every year, which is equivalent to about 30 gray whale carcasses. Because these carcasses come in a big pulse, first of all, we were able to measure the stoichiometry of carcasses by pulling fresh carcasses out of the river, dissecting them into their components, and then measuring the stoichiometry of those components. We've kind of recompiled, how much carbon, nitrogen, and phosphorus carcasses bring in. And then we could trace the pathway of those nutrient inputs into, as they move downstream. are consumed by aquatic and terrestrial scavengers and influence river ecosystem processes. And one of the things that was most striking, and you probably know a bit more about this now after seeing that awesome talk by Elizabeth and Judith, is that they have a whole lot of phosphorus in their bones. So we estimated about 95% of the phosphorus in a wildebeest carcass is in their bones, and those bones decompose really, really slowly. So a huge portion of those that phosphorus, as well as nitrogen and carbon, is just kind of retained over long time scales, over months and months, up to years and decades. Whereas the soft tissue decomposes really quickly, and that can result in a big pulse in dissolved inorganic nutrients, increasing like ammonium by 20 times and doubling nutrient uptake length downstream. So whereas it would maybe take 30 kilometers after a week for an ammonium molecule to be assimilated by the river, it can take double that after 2 weeks of having carcasses in the river. These carcass inputs also feed thousands of terrestrial scavengers. So this is a video showing all of the marabou storks and vultures of different kinds that come and feed on carcasses for several weeks after drowning. And we've documented some really interesting patterns of scavenger succession. And because the bones decompose over decades, we estimate time to loss at like 85 years, and the input rates are high and the decomposition rates are low. This leads to a really large estimated standing stock of about 5,100 tons of bones. So this is, every ton is 1,000 kilograms. So 5,100,000 kilograms of bones in the river at any given time. And as you can see on this bone here, bones also grow a biofilm over time, and that biofilm is richer in chlorophyll A and ash-free dry mass, so richer in organic matter and algal biomass than the biofilm that grows on rock. And that seems to provide an important food source that then can persist potentially as long as the bones do. So provide a food source that persists for decades after carcasses are gone. And we see a bit of this with stable isotope tissue of fish assimilation, where we see that when carcasses are present in the river, they're an important food source comprising up to 50% of assimilated tissue. But even months after carcasses have essentially disappeared from the river, all the soft tissue is gone, we see that the bone biofilm, which is isotopically distinct from rock biofilm, still is a really important part of their diet and actually potentially exceeds the importance of rock biofilm. And yet, despite all of these large and important ecological effects that carcasses have on rivers, this river, when we look at it from kind of a whole ecosystem scale with measures of whole stream metabolism, we don't see a big effect. There's no difference in gross primary production of this river when carcasses are present versus when they're absent. So 5,000 carcasses in a river has no effect on gross primary production. That is surprising. And we see actually a slight decrease in ecosystem respiration. So why is that happening? And we think it's because of the context in which those carcasses are entering. And that is, the animals are bringing in a really rich resource subsidy in terms of thousands of carcasses, but it's entering a system which has already been shaped by the inputs of 4,000 hippos. And those hippos increase turbidity, increase the carbon loading by 700% compared to upstream. They have a huge impact on both the physical and the abiotic and biotic community of the river system. And the carcasses are coming as a pulse into a system that's already been heavily shaped by this other animal resource subsidy. And Jonas's work, in the presentation, talked a lot about ecological effects of that. If you want just a visual for what hippos can do to an aquatic system, this is some work we did at the Milwaukee County Zoo hippo exhibit trying to quantify it. And you can see, as well as any graph can show, really quickly what hippos can do to a river. So I want to thank the many collaborators, my research partners with whom I've done this work, and collaborators across many institutions both in the US and Europe and Kenya, and funding sources who've made it possible. And thank you all for being here, and thanks to the organizers of this sessions. It's been really fun.
Nacho,
You're on mute, Nacho.
Thank you very much. Sorry. about this. Very interesting talk, and very interesting how hippos and wildebeest interact in order to enrich the river, but still clean it and subsidize also scavengers, etc., etc. We can talk about that later. I'm gonna just share my presentation, just because we're really tight on time, and we'll can discuss we have some time for quick questions later, okay? Just very fast. Okay.
Hello, my name is Nacho Vilar, and I'm gonna tell you about how large frugivores modulate the nitrogen cycle on tropical forests.
First, it is key to understand that tropical forests are loaded with fruits of different colors, shapes, and sizes. All sorts of animals feed on fruits on tropical forests. And actually, many large herbivores in tropical forests are voracious fruit and seed hunters.
Now, I don't blame them, Fruits are so good.
As they roam and search for fruits through the forest, large frugivores like tapirs and peccaries trample the soil and defecate throughout it.
Now, you would expect that trees and palms that produce large amounts of fruits will attract large frugivores, and in turn, this interaction between large frugivores and trees and palms would modify the soil, including the cycling of nutrients within the soil. And this takes me to the concept of fruiting lawn.
The concept of fruiting lawns is actually very intuitive. Trees and palms produce large amounts of fruits that attract large frugivores. In turn, large frugivores affect soil microbial communities through fruit consumption, dung and urine excreta, and physical trampling. This stimulates the activity of microorganisms involved in the nitrogen cycle, increasing nitrogen availability in the soil. Finally, plants capitalize on increased availability of nitrogen, increasing their food production, and here we go again through the positive feedback.
Now, to test the hypothesis of fruiting lawns, we capitalized on our long-term large herbivore exclosure experiment. So let me take you there.
The experiment consists of 42 large herbivore exclosures and 42 paired open plots where herbivores can roam freely, scattered through the 4 sites in the Atlantic forest. Now, as you can imagine, setting up an experiment of such magnitude was anything but trivial. For this particular study, we focused on one site where peccaries and tapirs are the largest frugivores. The fruiting king of the forest is the palm Euterpe edulis. In fact, we are accumulating growing evidence suggesting that palms like this might be foundation species across the Atlantic forest and also perhaps across the neotropics. But that's probably another story for another day, right? Today let's focus on soil nitrogen cycle. As you can imagine, in order to study how large herbivores affect soil nitrogen cycling, we use the latest high technology at hand. For starters, I conducted a pilot study using a utterly sophisticated gardening kit, gently provided by my boss at the time, Mauro Galetti.
Then we went for the real thing. We took soil from the paired open and exclusion plots and extracted nitrogen using an improvised lab. The fieldwork on the experiment is pretty hard work, but also lots of fun, especially when you got the best cook field technician in the Atlantic forest, the one and only Sergio Nazaret. Right, but let's get to the point. What did we find? For starters, we found that large herbivores led to a colossal increase in nitrogen availability. Right, and what about their effects on microbial activity? Well, we found that nitrification rates increase with increasing number of palms, but only on open plots. This suggests that large herbivores and palms together structure the activity of soil microorganisms. Right, so is there really a love story between these large frugivores and microorganisms? Well, actually, our results suggested this. We found, for example, that on open plots, microbial nitrification and soil nitrate concentrations were tightly linked, but that didn't occur on closed plots. This clearly suggests that large frugivores increase the performance of soil microorganisms. And now for the cherry on the cake. We found strong evidence suggesting that large frugivores, possibly through their movement, enrich or top up poor forest patches with low background levels of nitrogen, hence reducing the differences in nutrient load between more fertile and less fertile forest patches, and ultimately modulating the landscape-wide variance in nitrogen. So our proof of concept study went well beyond our expectations. We found that large frugivores increase soil nitrogen availability, increase nitrogen cycling rate, optimize the performance of soil nitrogen cycling microorganisms, modulate landscape level nitrogen availability. Okay, so I would like to thank all the funding bodies who made possible this research, especially FAPESP, and all the people who participated in the studies. It was a major challenge, but proved very successful. And thanks all of you for attending the talk.
He means all of you around the world!
I hope that you now better understand that large frugivores and the fruits they eat are not only cool, but also key to nutrient cycling in tropical forests. Right. So we're back. We're back to the session. I'm afraid, thank you very much. We are very tight on time. Huh? We are very tight. We don't have time. The organization is saying that in 1.5 minutes this is finished. So what we will do is to move the questions. I'm sure you got loads to the beers and nibbles that we organized from 5:15 p.m. Okay, we'll share a Zoom link there. I also pasted in the chat evaluation questionnaire that we would like to share with you. Please fill it in because we're thinking about the standalone symposium for the future, and we would like to know what do you think about today's session and the future symposium. It's a real pity that we don't have time for questions because I'm pretty sure that you got lots of interesting questions. But anyway, I just want to thank the organization, but especially all the speakers involved: Amanda, Jonas, Lisa, Judith, also Yuri and Claudia for helping, and all the audience for being here. And if you want to find more about Zoogeochemistry, just join us in our beers and evil session. Okay, thank you very much.
Also, we will share the answers and the outcome of the other quiz in the beers.
Oh yes, we'll
please join us. It would be so much fun.
Yeah, it's gonna be lots of fun and very distended.
Thank you very much, people of the world. See you in a bit. Bye bye.